All right. Good morning or good evening, wherever you are, to everyone, and welcome to the February 2026 installment of the GCE for All International Webinar Series, organized by the GCE for All Center and hosted by myself and my esteemed colleague, John Lewick. Here at this webinar, we feature scientists from all stages of their career and developing and using genetic code expansion, as well as experts in neighboring fields, including ribosome structure and function, tRNA therapeutics, directed evolution, and so on. And as always, if we'd love to hear from you folks, if you're interested or you know someone who would like to present, reach out to our center staff. Okay, so our first speaker today is Dr. Joon Lee, Associate Professor in the Department of Chemical Engineering at POSTECH in Korea. Dr. Lee is a chemical biologist and synthetic biologist whose work is broadly focused on engineering biological systems to generate new polymer types, including new classes of natural products, therapeutics, and precision polymers. Dr. Lee received his bachelor's in biology at Yonsei University in 2006 and went on to complete his PhD at the University of Oxford in 2015 with Hagen Bailey. He then carried out postdoctoral research with Michael Jewett at Northwestern University, where he gained expertise in cell-free protein translation and ribosome-mediated protein engineering. Shortly after that, he started his independent lab at POSTECH in 2021 and was promoted to associate professor in 2025. The central goal of Joon's research is to produce defined non-natural polymers using engineered biological translation, and he tackles these problems in three broad areas: expanding the number of fundamental building blocks available to translation, also engineering ribosome components and associated chaperones, as well as evolving ribozymes to attach non-canonical amino acids, having new backbone chemistries to tRNAs. Dr. Lee is published regularly in high-impact journals, including a recent Nature Communications paper that I think he'll be talking about today, where his team has demonstrated the ribosome can do more than form peptide bonds, and showing it can directly catalyze the formation of 5- and 6-membered cyclic backbones, actually doing it inside the peptidyl transferase center. So I'm super excited to hear a lot more about that. Dr. Lee, thanks for joining us today. Please take it away.
Yeah, that's wonderful introduction. And thank you so much for the invitation as well. I'm Joon Lee at POSTECH in South Korea, as reintroduced. And this is my great pleasure to be here on this global platform. My lab is 5 years old, or perhaps 5 years young. But our recent results have been really exciting these days, and I'm really excited to share them with the genetic code expansion community today. So what I like to discuss is how we can use the ribosome in cell-based systems as a catalyst to form a non-canonical cyclic backbones for biological polymers. So the backbones that I'm going to discuss in a few minutes differs from this macrocyclic backbone that you get from the post-translational glucations. So for example, like if you have one cysteine at the N-terminus of like a linear peptide, and if you have another another cysteine that is at the C-terminus, then they cyclize with the disulfide bond formation. That is the form, and that is the macrocyclic structures formed post-translationally. But what I'm going to tell you today is about the cyclic structures formed within the ribosome during the amino acid polymerization processes. So that is the point that I was just want to just quickly pointed it out. And this is my lab, and I currently have about like 15 grad students, and they are all incredibly talented. To be honest, I would be able to give this talk today without them. I'd like to thank my students for their hard work and their incredible ability to find clues or answers where there seem to be no answers. So we actually do pretty several different areas. So first of all, we develop new sulfate systems to incorporate non-canonical amino acid in the middle of peptide or proteins. And we also develop the sulfate system to incorporate those non-canonical amino acids more efficiently and more economically. I am not going to tell you about the new software systems, but this is uploaded on bioRxiv recently. So if you're interested in like what types of new software systems I am developing, please go to the bioRxiv and find my papers. I'll upload a link on one of my slides. Yeah. And then we also investigate the types of unknown chemistry that can be carried out outside of non-biological systems, and how we can utilize them to improve existing processes. Specifically, we are interested in producing high-value materials using this software system and chemistry. And then finally, we are also focused on synthesizing ionized lipids and non-canonical nucleic acid as well. Not only just non-canonical amino acid, we're also interested in non-canonical nucleic acids to do some funky chemistry using the software systems. Before getting started, I just want to tell you a short history of my PhD work. I was interested in incorporating a natural amino acid into the middle of a long peptide chain, which is assembled to a pore-forming protein. So this is basically a prototype version of the Oxford nanopore technology. What's the name of the devices? Yeah, MinION. This is one of the prototype proteins that was used for the MinION initially. But now they don't use alpha-hemolysin, but they use a CSGG protein for DNA sequencing. But anyway, when I was interested in incorporating a non-canonical amino acid into the middle of the alpha-hemolysin, I used a semi-synthetic approach. What that means is we synthesized this short peptide chemically, basically a solid phase peptide synthesis approach. And then we basically couple every single amino acids, protecting reactions and deprotecting reactions and coupling reactions, and those kind of repetitive and irregular processes, you can actually get a little peptide that contains a non-canonical amino acid in the middle. And then we synthesize these two long fragments from E. coli, and then purify them. And then when you synthesize this C-terminal fragment, you have to locate N-terminal cysteine at here. So the cysteine goes to here at C-terminus, or basically the thioester of this short peptide. And then the thioester furs up and forms an amide. And they are covalently linked together. So these reactions are called native chemical ligations. And using these two steps of native chemical ligations, I designed a one full-length monomer of alpha-hemolysin that only contains one non-canonical amino acid. Well, I love the work, but this was really hard. The interior membrane proteins are notorious for the solubility. So if you do if you do not use any detergent in the solution, then you basically all the proteins are just get precipitated out. Well, anyway, that was my PhD work. I was interested in incorporating non-canonical amino acid into a peptide or proteins. And the approach that I was using at the time was just chemical synthesis or semi-synthetic approach. But I think everyone here in this genetic code expansion knows about this strategy pioneered by Peter Schultz or Ribosomal. Amber codon suppression is a great tool to incorporate non-canonical amino acid into proteins or peptides. But as a point of view of synthetic biologists now, myself, I view this in vivo system has multiple limitations. So one of the limitations is that if you design a non-canonical amino acids that are not compatible with the membrane, basically if it doesn't get into the cell, because if it's like too large or if it's too hydrophilic, then the nucleic acid that you designed did not go into the cell membrane, did not go into the cellular materials like uh, cytoplasm. And also, a lot of people are confined to use the only amber cotton to incorporate non-canonical amino acid. But these days, polypeptides are also an alternative way to incorporate multiple non-canonical amino acids, but mostly using the amber cotton is kind of like limitations to incorporate non-canonical amino acids. So you're not allowed to incorporate multiple non-canonical amino acids easily. And one of the biggest challenges that you want to incorporate non-canonical amino acids using this in vivo system is that you need to develop a new orthogonal amino acid synthetase and tRNA pairs for the new non-canonical amino acids. And if it does not accepted by your orthogonal amino acid synthetase, you basically restart the re-engineering orthogonal tRNA synthetase, and then make library and select one of winners and test it out whether it accepts your non-canonical amino acids or not. And if it does not, then you have to restart, and so on and so forth. So this has a lot of limitations in point of view of the synthetic biologics. So we need a new way to incorporate the non-canonical amino acids, and the sulfur system is, from the point of my view, can be an efficient way to resolve these limitations. Because the systems do not contain cell wall, as its name tells, and you do not have any physical barrier to get to the biomaterials which you want to utilize with. Any non-canonical amino acids that you design can be at least introduced into the cellular material. For example, like E. coli have two types of cell-free systems. One is just simplest system. A lysate-based system. This is the system where you grow cells and lyse them and centrifuge at a certain speed and just take the supernate them and add like energy sources or amino acids. And when you add a DNA template that you want to synthesize, then it's transcribed by the polymerase, and this is already present in the cellular materials, and then translated into proteins by the ribosome and the other translation machinery, which was already present in the cell. Another system that we actually use for this study is a reconstituted system, or so-called pure system. This system only contains 36 different purified enzymes and purified ribosomes, and the purified tRNAs and purified translation machinery in it. And this system allows a cleaner synthesis of a particle. So all the studies that I'm going to show today is based on the pure system from NAB, but recently, as I talked at the very beginning of my presentation, we were also able to construct our own reconstituted system using an automated platform. So as I just mentioned, this was uploaded on bioRxiv a couple of months ago. So please find the literature if you're interested in how we actually construct the reconstituted sulfur system. So anyway, let me just get started. So from the experiences that I have so far, I feel using sulfur systems are the most efficient way to incorporate non-canonical amino acids. And the question is, how do we charge these non-canonical amino acids to tRNAs? The strategy that I'm using is the Flexism, which is a ribozyme that Hiroaki Suga at the University of Tokyo developed around 20 years ago with Jack Schuster. So if you imagine how AARS charges charges this amino acid phenylalanine to tRNA. It recognizes half of tRNA structures, and it also recognizes 3D structure of phenylalanine. Excuse me. Phenylalanine, and then it couples the phenylalanine to the 3' hydroxyl group of this tRNA. But since we are not using ARS on the sulfur systems, we just use the synthetic substrate, which has an ester at the C-terminus. So if you compare these two molecules, this is an acid and this is an ester. So the only caveat of using flexizyme is that you need to synthesize a non-canonical amino acid and ester. But if you can synthesize it, you just mix them together with tRNA. And then if you add a flexizyme, and this recognizes the 4 end sequences like ACCA, and complementary forces. And then this hydroxy group attaches here, and then the phenylalanine actually is charged on tRNA. So if you compare these two consequences, it is exactly the same. So you don't really need to use ARS to charge a non-canonical substrate or canonical substrate to tRNA. So if I summarize what I just told you, since the cell carries out the transcription and then translation to incorporate a certain amino acid into the middle of peptide or protein, but since the sulfur system does not contain any physical barrier, we just use the ribozyme or flexizyme to charge this non-canonical amino acid to tRNA, and then just spike it into the sulfur system so that all the other translation machinery delivers it to the ribosome, and then ribosome accepts it in A site, and then it polymerizes the non-canonical amino acid in the P site, and then tRNA exits through E site, and then if this is folded, then you get a proteins or peptides that contain non-canonical amino acids specifically. So that's the key idea. So we have been synthesizing a lot of different non-canonical amino acids for a long time, and it turns out, to make a long story short, it turns out that there are actually rules. So one of the rules that we follow is that if you design a non-canonical substrate similar to phenylalanine, basically you have carbonated carbon and you have an ester with a living group, and if we have 2 carbon linkers and an aromatic group, then flexizyme loves that it just attaches it to the tRNA. And if you have electron withdrawing group on the substrate, then the electron withdrawing group pulls down the electron from the carbon, then it makes easier for tRNA to attack the carbon, so it can actually facilitate the tRNA acylation reaction. But if you design electron donating group on it, then basically it pushes the electron to the carbonate center, so it basically slows down the flexizyme charging reaction. And as the same manner, if we have a bulky substituent or bulky moiety around this carbon, then it makes harder for tRNA to attack the carbon, so it basically slows down the reaction. So by following this design rules, we were able to expand a lot of the range of the substrate that can be compatible with the flexizyme to a significant spectrum. So for example, we were able to charge any alpha amino acid and also with a D amino acid, like with the mirroring form. And then we were also able to charge like beta or gamma or all the way to the zeta amino acids. And we were also able to charge like fat amino acid, like with 4-member ring or 5-member ring or 6-member ring. Then we were also even charged like fluorescent dyes based on the alpha amino acids stem into any of peptides or protein chains. So virtually any amino acids can be acylated into tRNA if they are appropriately protected or deprotected during the chemical synthesis of small non-canonical substrates. So the idea is that we design non-canonical amino acid, we charge non-canonical amino acid to tRNA using glycoside system. And then if we have a multiple tRNAs that recognizes multiple different codons, then we just spike it into the self-lift system so that it goes in either N-terminus, C-terminus, or in the middle of the peptide, or in multiple positions of proteins. So that's basically what we are doing. That's the key concept. Okay, so switching the gear a little bit, by the time we are synthesizing lots of different non-canonical amino acids, we were inspired by the structure of the natural products. One of the structure that we mostly inspired on was penicillin. Well, I think everyone knows here, everyone on the earth has an experience of taking penicillin, I think. But they did not know the structure for sure. But if you look at the structure, the molecular structure here, it is a valine. So this is valine. And if you look at this way, this is cysteine. So for somehow, like within the mechanism of the cell, where exactly the non-ribosomal peptide synthetase fuse those two amino acids together into a ring structure. And if they contain this cyclic structure, then it gets a drug effect. This telamine or godsprin also contains a lot of multiple rings in it. It looks like serine, it looks like cysteine. If we have a way to couple together with somehow, or using different biomaterials or enzymes or chemical way, then we can actually build these drugs or pharmaceutical products using the software systems or other chemistry. So our question was, can the ribosome produce these types of cyclic backbones? So to answer the question, we actually went back to the high school chemistry and then tried to look at what actually happens within the ribosome. This is the amino acid located in the P site, and this is the amino acid. I'm sorry, this is the amino acid located in the P site, and this is the amino acid coming into the A site. And the amino group of amino acids on the A site attacks the carbon that is connected to tRNA. And this amine attacks here, it forms amide and tRNA falls out. So if you look at this build, if you look at this picture, ribosome is actually a machine that has evolved for a long time, millions of years or billions of years, to only attach the amino acid with an amide bond. If I teach organic chemistry in a classroom, I teach this reaction as aminolysis. So ribosome is basically the chemical machine that carries out the consecutive aminolysis reactions over and over for a long time to make proteins or to make life or to make humans as well. If I simplify the mechanism that I just showed in the previous slide, this is a nucleophile. The amino acid in A site is a nucleophile, and the amino acid in the P site is actually electrophile. And this is basically nucleophilic addition reaction into the electrophile, and it forms a covalent bond between electrophile and nucleophile. So our idea is that if we design a non-canonical amino acids with two nucleophiles, then if we could also design a non-canonical amino acids or non-canonical substrate with two electrophiles, would they form a ring structure? For example, if this nucleophile attacks here, if this nucleophile attacks there, so form a ring structure. That was our question. So we designed the actual molecule to answer the question. Like we design hydrazine, which contains two nitrogens. So basically, this is one nucleophile and this is a nucleophile. And then we also design a gamma keto ester. So it contains a ketone at the gamma position and also an ester. These are the two representative electrophiles in nature. So if these two motifs come together within the ribosome, we form the six-member ring. That was our actual question. It turns out that this motif are a very usual motif to produce a drug efficacy. So for example, these two small molecules are drugs that are used for a asthma or chronic diseases. But we think that if we could synthesize this the pharmaceutical motifs using the ribosome, that would convert a paradigm of this chemical synthesis. So we designed these four different gamma keto esters, and then we also designed these two different hydrazines, and then we mix them together into software system that contains ribosomes, other endogenous tRNAs and amino acid. And within the reaction, within 2 hours, if you don't really need to use organic solvent, and under the mild condition, like at the 37 degrees Celsius, we were able to form 8 different PRZ rings by just playing around with different gamma keto esters and different hydrazine substrates. So that was the first observations that the ribosome forms a cyclic motifs rather than just like linear peptides. So we were quite excited about this result, but we had still lots of remaining questions. So one of the questions was, can the ribosome produces other types of rings, for example, like a 4-member ring or 5-member ring or 6, 7-member? And also can the ring closing reaction routine, can you just leave the reaction open so that the ribosome just stops in the middle and it just produces a linear structure rather than just this closed structure? And another interesting question was, I think some of you might already notice that, how do you know that this alpha nitrogen goes to here and beta nitrogen goes to there, rather than just beta nitrogen goes here and alpha goes to there? So to answer those questions, we designed another set of non-canonical substrates that can be used for new cyclic backwards. So this is basically 3 carbon chains derivatives, malonate acid derivatives. And this is 4 carbon chain derivatives, like 1, 2, 3, 4 carbon chain derivatives and succinic acid derivatives. So we designed these 14 different substrates as a ring closing motif. And then we asked whether this formed different types of ring structure, like 5-member ring or 6-member ring with 2 carbonyls. So this was the previous work that we were able to report in 2022. And these two mechanisms are the new mechanisms that we were able to report in 2025, which is last year. We designed 2 ester substrates, basically based on the succinate ester. And we were playing around with this R1 group as well. And if we also decreases the carbon chain length by 1 carbon, then you will form a 5-member ring. And if you just use the 4 carbon substrates, then you will also get a 6-member ring, but with a slightly different structures with 2 carbon. So that was the idea. It turns out, to make long story short, I think I'm running out of time. To make long story short, we were able to form nine different or three different five-member rings and six different six-member rings as well, using exactly the same strategy. So the concept is that we also play around with the R1 group as well. If R1 is a good living group, then this ring formation will be faster, will be accelerated. Or if this R1 group is a poor living group, then the ring formation will not be accelerated. And as a result, you will get a linearized product, as you can see here. So those are our idea. And it turns out that if you look at this mass spec, five-member ring was the most stable ring found in the nature, and we will also form this five-member ring pretty easily by just playing around with the R1 group. But the six-member ring was interesting. If we design a four living group at the R1 group here, as I just mentioned, then you can slow down the reaction completely. So you get only just a linearized product. But if you switch it to a better living group, then it actually facilitates the chemistry within the ribosome. Then it forms it facilitates the ring closing reaction really rapid. For the residual selectivity issue, we designed this linearized peptide. So we basically designed a succinate ester which contains a full living group, and then we also designed this hydrazine ester. And then when it couples, it forms a linearized product by just 80% of the total amount. That is because this methyl group is not a good living group. So 20% are 20% of the total product is about the closed react is about the closed structure, and the other 80% is just a linearized product. If the linearized product forms like this, then when we add shift base to there, then it will form a material like this. Then if this is formed, then the alpha nitrogen goes to the tRNA ester, and then beta nitrogen active is not the one that attacks the tRNA. It's still active as a ring closing the atom. So by adding just the sheet base to the system, we observe that the total peak is completely shifted to the corresponding mass that we imagine is theoretical masses. So we were able to conclude that this alpha nitrogen goes to the tRNA, it forms an amide force, and then the beta nitrogen goes to the other ester to close the ring. So we were able to propose a
real quick, June,
mechanism.
you're kind of going over time here, so let's hit the high points
Okay, so this is the last slide. I'm sorry. So this is the last slide. Yeah, so we were able to propose a mechanism. Alpha nitrogen goes to here, and beta nitrogen closes the ring. And those are basically what we have observed. So to conclude, ribosome has evolved to polymerize amino acid into a protein, but in our lab, we use the ribosome as a catalyst, and particularly we are interested in forming a five-member ring or six-member ring or seven-member ring, perhaps, using this non-canonical substrates. Thank you very much for listening to my talk. The five-member ring and six-member ring was done by Joongoo, who's here in the gold t-shirts, and the sulfur systems are basically done by Byung-min, who's here. So, yeah, thank you.
All right, thank you very much. If there's people who have questions, you can put them in the chat, and we'll do our best to get to them. Otherwise, we can have you ask questions in person. I think we have the power to do that. I have a quick question for you, June. So you've really done some beautiful work to show that you can make these cyclic peptides using ribosomes. How do you foresee, I mean, and your efficiencies look pretty good. But what about yield? So what would it take to scale this to be used for, you know, a therapeutic or whatnot?
This is a great question. And this is the question that I always got from the conference when I just give talk. Well, one of the biggest disadvantage of the sulfur system is that it does not give enough products. But that's just conventional and traditional problem with sulfur systems. And we are using a microliter scale, and you get only nanogram of the peptide. But it's simple. If you want to like increases the yield, you can basically increase the volume to milliliter. But the problem is that it's too expensive. For example, a pure system is really expensive. It costs like $500 per like 20 reactions. So you were not able to incorporate. So the system that we uploaded in the bioarchive is how to produce like economic sulfur systems, reconstituted systems. So hopefully that helps to increase the yield of peptide or proteins that is produced in the sulfur system.
And it's another question. I'll ask one more quick one. Do you ever foresee a flexozyme system where you can make it specific enough to a suppressor tRNA that you could enable its use inside cells?
Yeah. This is really interesting question. And I haven't I have thought about this question before, but this is really hard. I mean, the flexozyme is only 45 nucleotide long, and if you produce it inside a cell, then it will be chopped up by the protease, the nucleases. So you can't use it inside the cell. There is a different models that modifies the length or the sequences. For example, like T-Rex system. So you can basically use it inside the cell, but you can't actually incorporate non-canonical amino acid inside the cell.
Got it. Great. Well, thank you very much, June.